In Sweden, Norrbotten, the northernmost county, there is a city that is so isolated that if there was a bad harvest one year, people would die, but a good harvest another year, people would feast. For the people who grew up during famines, they would live an average of 6 years longer than those who grew up overeating. But that's not all. Not only did the people who grew up during years of feasting live shorter lives, but so did their children and grandchildren, and it was the same in the opposite way for those who grew up during famines. This is all due to epigenetics. Genes code for proteins, which can control our phenotype, or the traits we possess. Most people think of DNA as just bases being read, leading directly to proteins, but there's actually a lot more going on. One very important aspect of DNA translation is epigenetics. Epigenetics are sort of like the asterisks and highlights and extra markings on a page that we add, which adjust the meaning in a seemingly small way, but actually really change the traits of the hosting organism. Epigenetics are heritable traits that cannot be explained by changes in DNA sequence. It often refers to changes in a chromosome that affect gene activity and expression. Epigenetics is the reason muscle cells, neurons, skin cells, and all of the different cells in your body functions differently, despite having the same genome. Epigenetic traits can be picked up and change over one's lifetime, so this means that different habits or compounds you are exposed to over your life can affect the expression of your genes. Since epigenetic traits are heritable, it can also affect your children or grandchildren's health. The most important thing about epigenetics is that it controls the output of genes without actually altering the content of the genes themselves. One of the mechanisms of epigenetics is methylation. Methyl groups are little molecules similar to methane that are very common in biology. DNA methylation is a process by which methyl groups are added to the DNA molecule, and two of the bases of DNA can be methylated, cytosine and adenine, but adenine methylation is much less common in mammals. Methylation results in the gene it is on being unreadable, effectively turning it off, like a switch. In cytosine, methylation often occurs on the fifth carbon of the cytosine base ring. A common configuration of cytosine methylation is where a methylated cytosine is adjacent to a guanine, whose complementary cytosine is also methylated. This is known as CpG methylation. CpG methylation results in the gene it is on being unreadable in both directions, which effectively removes the gene or only activates it under certain conditions. Methylation can be affected by different habits, and bad diets and smoking can make methylation mistakes more common, which not only affects your health, but also your children and grandchildren's health, because not all epigenetic tags are removed whenever a new generation is created. Another aspect of epigenetics is histones and their modifications. Histones are proteins found in cell nuclei that keep DNA in a contained order. They keep and contain DNA by wrapping the DNA around themselves, and nucleosomes are clusters of histones that act as a spool of DNA. The existence of nucleosomes shorten the length of our DNA by a factor of 10 million to 1, and structure and organize the chromatin in the cell nucleus, especially during mitosis. They also mark the locations of ultraviolet damage to lessen the potentially harmful effects of mutation. Histone modifications are a separate process from methylation, where many different codes instruct how each histone behaves, such as winding the DNA around the histones tighter or looser. Histone codes contained on their tails can even be methylated. Despite this, histone modifications are not completely understood. When DNA is tighter, it is less likely to be read, and when the DNA around histones is looser, it is much more likely to be read. Because of this, histones can affect how often genes are expressed, and by extension, how common a protein in an organism is. This is why histones are similar to a volume dial, making proteins more or less common. However, histone modifications are different to methylation because while methylation simply turns a gene on or off, histone modifications affect gene expression on a gradient. So a certain protein for keratin, for example, might be more common in a cell that makes hair than a cell that makes our teeth. But teeth-making cells still have keratin, so this could be controlled by histones, along with many other cellular examples. Histone modifications are also useful because of some new proteins may only be useful during a certain stage of a cell or at a specific quantity throughout a cell's lifespan. Today we learned about epigenetics, that it is a vital process to DNA and life's existence. We learned that when DNA methylation occurs, it can be likened to a switch turning off or muting that gene. Histones, however, act as dials, which control how often different proteins are produced, as well as a variety of different functions depending on what kind of histone it is. I'm Adrian Chapman. Thanks for watching.